Good morning, my dear friends. I am Palak Vyas from Indukaka Ikkovala College of Pharmacy, the CVM University. Today, I am going to share with you some knowledge regarding the X-Pharma software for all these pharmacology practicals. As we know, why did pharma software come into existence? Traditionally, pharmacology practicals involved experimentation on live animals, for example, frogs, rabbits, guinea pigs, rats, to study the drug effects on tissues, organs, or whole organisms. However, the use of animals in practical teaching faced significant challenges regarding whether it is about ethical concerns, some legal restrictions, or some practical constraints. So, to overcome these conditions, an ex-pharma software was developed by the Bureau of Health and Education. So today I am going to show how to operate the X-Pharma software. So here you can see the hapnic.in/xpharma software website here. So, you have to log in via the institutional login ID and password. So here it is written, this is the software which shows the X-Pharma pharmacological experiments, which is the computer assisted learning simulation software for the students of uh, pharmacology subject. So here, first of all, you have to enter in the institutional panel of the X-Pharma software. So you can see the diagram of the experimental animal, that is albino rat. Now you have to enter the password, ID, username and the password of provided by the Bureau of Health and Education to the institution, to your respective institution. So here I am entering my username and I'm entering the password. Okay, now you have to click on the login button. So now guys, you can see there are different list of experiments coming under the pharmacology practicals in semester 4, 5 and 6. So there are various list of the practicals you can see. But before starting the demonstration of this any one practical, I would like to suggest some information regarding the X-Pharma software. So how one can benefit or the students can benefit from the X-Pharma software. So first one is regarding the students can gain the practical knowledge without compromising any animal welfare, because considering the ethical considerations, CPCAC guidelines and the UGC guidelines, and as well as the animal welfare and rights have made strict regulations regarding the usage of the animal at the bachelor's level. Also, the software provides the stepwise procedures, graphical data, and animations, enabling better visualizations of various drugs action. So the student can easily understand the type of practical. They don't have to run on for different preparation of the dosage forms and everything. So they can easily learn via this simulated kind of the practicals. Students can also repeat the simulations multiple times, reinforcing their learning without limitations of the animal fatigue or the tissue damage. So this is regarding the obstacles coming during the practical work. So we don't have to deal with any of the animal considerations during the practical work. Also, the any experiments can be conducted quickly, anytime and anywhere in the computer labs, overcoming some logistical constraints. So, despite of dealing with the animal obstacles, we can easily access this software anywhere, any place, at any time, and with the quick responses. Also, it helps in revising practical procedures before the exams, building confidence and accuracy in explaining the experimental pharmacology. So the students can easily understand the practicals if they are not aware regarding any single practical. They can easily operate this software and they can understand all the procedure as well as principles, observation, results regarding the specific type of the practicals. And also, it leads to exposure of various tools which make students familiar with modern approaches in the pharmacology education, which is important for their future research and the professional standard. As we know, AI technology has been implicated everywhere in the education system and all the areas of research and development. the student nowadays, the students are more inclined towards the AI technologies. So this is one of the example of the usage of the AI technology by the students. So they can easily operate this, and it is beneficial for their future research and professional work. So thus, in the end, I would conclude that X-Pharma software is emerged as a replacement and supplement to the animal experimentation, aligning with the 3 R's principle. That is regarding the reduction. Reduction here means the reduction in the usage of the animals, replacement. So animals are replaced by the AI technology, which is very beneficial when you are considering the animal welfare and rights. And refinement. It enhances learning outcomes and which ensures ethical practices and equip pharmacy students with updated skills for their clinical research and the academic excellence. So now, this was regarding the Experima software. Now I would demonstrate one experiment so the students and the related person in the field of pharmacology can easily understand how to use this simulated kind of the practical. So you here there is a list of the experimental pharmacology series you can see, that is, for example, the study of muscle relaxant activity with the help of rotarod apparatus, study of CNS depressant and stimulants using actophotometer, study of the analgesic activity with the help of tail flick apparatus, study of various histaminic drugs with the help of histamine chamber, that is the example of the mast cell stabilization method. The other one is the study of analgesic activity with the help of hot plate apparatus, study of drugs on CNS using the elevated plus phase apparatus. So these all, and also the study of anticonvulsant activity by electroconvulsometer. So these all experiments are regarding the pharmacological instruments which we are using for studying the various effects of the drug. For example, whether it is about the antihistaminic drug, whether it is about the CNS depressant drug or anti-analgesic drugs or some CNS or some muscle relaxant activity like drugs. Also, we can study the effect on various drugs on the individual isolated organs or whether the whole body of the animal. So there is experiments regarding this effects of various drugs on the rabbit side, to study analgesic activity by breathing test, to study it is at induced convergence in the mice, effect of different drugs on the frog's heart, recording of the DRC and bioassay practicals are also performed here. So the students can easily understand the assembly which is set up in the pharmacological lab, how to operate it. Okay, so that is also very well understood by these modules. Experiments of red blood sugar level, also basic bioassay practicals, the 3-point method, 4-point method, and the matching and as well as the interpolation method, all methods which is not feasible to conduct in the laboratory nowadays. So it can be easily understood by the students in a well explained manner. Also, the various study of the diuretic drugs using the metabolic age, some anti-inflammatory activity, some right-bit pyrogen test, various effects of the drugs on dog blood pressure and heart rates, study of anti-ulcer activities, study of various catatonic activities, and etc. Also, the skin irritation test is also being studied by help of these kind of the modules. And one thing I would like to notify that there is an updation in this Xpharma software on the regular basis. Either it is monthly or 6 monthly basis. So all the experiments which are coming under the syllabus of under the PCI syllabus of semester 4, 5, and 6. So almost all the practicals are included in this software. So there are also different 1 or 2 versions regarding the Xpharma software. So based on your study purpose, you can subscribe that whether it is a comprehensive pack or simple uh, neo pack for your institutional educational work. Also, not only the practicals, they are providing supportive resources for how to give different routes of drug administration, so the students can gain the knowledge of different how to administer various drugs at different routes of the administration for the laboratory animals. They are also teaching the various blood withdrawal techniques for all the laboratory animals. Also, they are explaining different methods of how to use anesthesia and euthanasia for the laboratory animals for performing various practicals. And also, they are explaining about the dose calculation in various pharmacological experiments. So, these are the methodologies which are done prior to the experimental work. So, this also is explained by this Experma software. Also, there are various visual models for some common laboratory animals. So we can gain, students can gain the knowledge regarding which are the laboratory animals used in the pharmacology lab. Also, the type of physiological solutions which are used in the experimental pharmacology, and also various instrumentations. They also explains about the various types of the instruments used in the pharmacology practical. Also, there is one option for the exam mode for the practical performance. So, if the respective faculty is more inclined towards this software, they can also perform the practical exam via this exam mode also. So, there is a very well understanding of the practicals. Also, there is some additional teacher-student reporting panel, so one can give remarks or one can give suggestion in this Xpharma software. So I hope you are very well aware with this Xpharma software, which is very much important for the studying the simulated practical in the pharmacology field. Okay, so now we will start with the example of one of the practical, so the students or the viewers can easily understand how to operate this software. So we will study about the study of CNS depressant and stimulants using the actophotometer. Okay, so we will click on this play button. Now you can see the screen. There is written the aim, that is study of CNS depressant and stimulants using the actophotometer. So there are two options given here, that is regarding the actophotometer and virtual practice actophotometer. Virtual here refers to the virtual lab. Okay, so we will click on to the virtual practice actophotometer. Now there is one option highlighting to start the experiment. We have to click here. So I'm clicking here, and you can see the instrument which is used for studying the CNS depressant activity. This instrumentation is known as the actophotometer. All the pharmacology labs at the BPharm and InPharm levels have this. All the institutions are having this actophotometer for studying this type of the CNS depressant activity drugs. You can see there is written the description of the instrument. So it is written that actophotometer has a central chamber with, you can see here the central chamber with arrangement of light sources and photocells at the base of two opposite walls. So these walls, you can see there are different photocells arranged. Both are focusing on the laser lights. The light of each source is focused on a photocell. Any interruption in the path of the light activates the photocells, and this is counted as a measure of horizontal locomotor activity of the mice kept in the chamber. So here experimental animals used as the mice. We are using the mice, or either you can use the rats also for performing this practical. Okay, so it is regarding the how the instrumentation works, or what is present in the pharmacological this instrumentation. uh, Now here there is one option of the principle of this actophotometer. So you can see the principle of the actophotometer. Here you can see there is a mice placed on the rods. So what is the principle behind this experiment? So the drug action on central nervous system as stimulants or suppressants affect the locomotor activity in the experimental animals. So when we are using the rats or mice without giving any CNS depressant drugs, so they will having a higher amount of the locomotor activity compared to the animals which are given the CNS stimulants or the suppressant drugs. So that we have to compare in via this pharmacological equipment. Now, such changes in the motor activity can be easily determined by using simple techniques in which the animal treated with drugs is kept in an open compartment having square marking on its floor. So you can see here an animal is placed here. The lines of such square crossed by the animal are counted by the observer. So the rat here or the mice you can see here will move here and there. Okay, so now there is a placement of the photocells at both these compartments. So when the mice moves here and there, the photocells, the beam of the light of the photocells will get cut off, and that markings or that cutoffs you have to measure at this digital panel. That cutoffs has been recorded, and you have to mark the number of cutoffs done by the experimental animal. Alternatively, the locomotor activity can be determined using actophotometer, or more sensitively by using automated video tracking system. In addition to the horizontal movement, the vertical movement is also considered as a parameter associated with the locomotor activity. So, what this line suggests that you can easily directly measure the cutoff time, or you can measure the horizontal movement and the vertical movement of the mice. Either way you can opt for it. So this was about the principle of the actophotometer. Now the next option is called the experimental work. So we will click on the experiment option. So there is instructions, or you can say the procedure given for the experiment. Okay, so we have to take animals into two groups, and we know each group will be consisting of 6 animals. Now we have to administer one group with the drug to be tested and other with vehicle by other route. Vehicle here refers to normal saline solution we are giving, and the one group is given the CNS depressant drug. Now put one animal at a time in the actophotometer. So one by one you have to place the animal, whether it is of the control group or whether it is of the drug treated group, one by one into the actophotometer, and you have to mark the observation. Now you have to start the instrument, count the locomotor activity for specific time. Locomotor activity refers to the here and there movement of the experimental animal. Now repeat the procedure at the interval of 30 minutes. Record the observations. The mice in test group are injected diazepam, that is a CNS depressant drug, with a dose of 1 milligram per kg or orally. Per kg refers to the weight of the animal. So according to the weight of the animal, you have to calculate the dose of the drug to be given to the experimental animal. So here the dosage form we are using, that is 1 milligram per kg of the mice, and via the oral route. And locomotor activity is measured twice as stated above. So the locomotor activity is measured at the end. So this was regarding the how to operate or how to do the procedure for performing this particular experiment work. Now we will start the experiment. So here you can observe on the screen there are animals. How much animals are taken? 12 animals, 12 mice is being taken, is considered for the pharmacological practical for this thing. So one group, we have to divide into two groups. The one group is the vehicle treated group, and the other group is the drug treated group. Here there is one button depicting administer, and here the vehicle substance is there, and here the drug, that is the diazepam 1 milligram per kg solution is being used. So now when you click on the administer button, you can see here how the uh, drug is being administered to the experimental animal. It is via the oral route. So this way you have to hold the animal and you have to ingest the drug via the oral route, and you have to place the animals. Now you can see the 6 animals are divided into the vehicle treated group, and 6 animals are divided into the drug treated group. So now what you have to do, you have to click on the individual mice and the experimental work will start. So I'm clicking on one of the mice for the vehicle treated group. You can see in this video, you can see the photo cells here. You can see the mice moving. It is this movement is called as the rearing procedure, rearing done by the mice. And here you can see the digital panel wherein the movement or the rearing is being measured. And here in this table you have to write the vehicle rated locomotor index and the number of rearings. This represents the locomotor index or the number of rearings done by the experimental animal. You can see here the numbering is being mentioned here, so we can easily visualize if you are not able to visualize on the video screen. This way, one by one, you have to select the animals. For example, this one is the second animal. You have to note down the reading again. Then you select the third one. Then you select the fourth animal. Then you select the fifth animal, and then you select the sixth animal of one group. And the readings you have to note down. Afterwards, you have to jump on to the drug treated group. So I'm clicking on the drug treated group, and you have to observe the difference between the cutoff time or the locomotor time of the drug treated group. You have to first of all write the observations. You have to click again on the second animal, the third animal, the fourth animal, fifth animal, and the sixth animal. One thing I would like to notify that all the drugs have been given the prior drug treatment before doing the experimental work. So before starting or dividing into two groups, okay, we are giving the vehicle and the drug to the each group of the animals, and then onwards we will put the animals one by one in the ectophotometer, and then we can get the observation table. I hope the procedure is clear to you. Now we have to click on the observation table, which shows this tabular form, which is showing the control group, the drug treated group, and the percentage decrease in the locomotor index. So here you have to note down according to that cutoff time. You have to note down the number of rearings, and the number of rearings means the forward and backward movement done by the animals. So that you have to note down, as well as the locomotor index, which is mentioned on the panel. So this you have to represent via with the help of this control group and the drug treated group. In the same manner, you have to note down, and you have to calculate the percentage decrease in the locomotion, because the drug we are using, that is the CNS depressant drug. So what will be the our result means the drug treated group will be having the lower locomotor index and lower rearings compared to the control group. So the percentage will be in a decreased form, and you have to calculate the average, and you have to gain the conclusion. You can, if we are going for the exam module, one can write the exam name, or the sorry, the student name of the particular student going for conducting the exams. Okay, and you can also save and download the observation table for your laboratory manuals. Now the experimental work is done, and finally we can get the conclusion. You can click on the conclusion button here, and what will be the conclusion? The observed reduction in locomotor index shows that the given test drug, that is the diazepam, exerts inhibition of the locomotor activity. So this will be your conclusion. Either you can write this or mention this in your laboratory manuals, or it is also beneficial for your examination purpose. So this health education and bureau, the Bureau of Health and Education System has modified this all experimental via the AI technology in a very well explained and detailed manner, so that you can easily understand this kind of the simulated practical. So here, so this was one of the example of this XPharma software. I hope you have very well understood how to operate or how to use this XPharma software for all the pharmacological experimental work. So I hope you are all clear with a short and brief demo of the XPharma software. And if any queries regarding this XPharma software, you can refer to the Bureau of Health and Education. You can notify them via email, via WhatsApp, or via institutional letter. So here there will be your institution name particularly for each individual institution. Okay, so hereby I am uh, finishing off with this small demonstration of the virtual lab techniques or the XPharma software demonstration practical. Thank you and have a good day. Jai Hind.